I am going to talk to you about tools today, technical tools. And I will talk about assessment of ecological continuity. As an introduction, I would say we can say, why are we looking to assess? Well, there are different objectives, whether we look upstream of the project or after the project is finished. The first point is to check whether we reached the objectives. That seems logical, doesn't it? Fair enough, especially for public wide-scope projects. The second point is to get some data to inform, raise awareness among all stakeholders, officials and wider audience. The point of this assessment is also to improve using experience, feedback, local feedback with different time and space scales. And the point is also to generate objective knowledge, generate debate, and in the end, try and get our policies in the right direction and help in decision-making process. So in order to do that, we need to gather concrete elements which are measured in the field, in situ. This becomes complicated because around ecological continuity, there are different compartments, different segments we need to assess. And as you all know, to get some knowledge on all these compartments, we need a lot of funds. So it's a sort of iterative circle because we get some data which will help us prioritize future actions, and future actions will help us define future objectives, help us find out whether they are achievable. And this is my introduction. So everything I am going to present is just regarding one area, the fish fauna continuity. So of course there are other compartments we need to include. So what do I have to present regarding fish fauna continuity? Well, we have different tools today. We can look at fisheries, we can look at traps in the fish passes, we can tag, follow the presence. We can do a bit more complicated things like otolith microchemistry and remote radio and acoustic telemetry. And I'm going to present three possible tools: the population genetics, the pit tags and RFID, and remote counting. I don't have a lot of time today, so that's why the third point is about remote counting, video counting, which I'm not going to present, but there is a poster in the hall, which you can then consult and ask me if you have any questions at the end. So we have a wide panel of tools, as I said, and we have to take into account the time scale, the space, see which species we want to target, which step of the project are we in, what budget do we get, what are the technical skills we have, which type of environments and barriers do we work on. And the main question is, what is the information we're looking to get? And then it would be relevant. Which tool will be relevant for which type of information? So the first tool I would like to mention today is RFID tool, which is really robust, efficient, to check the efficiency of fish passes or fish crossing tools. So I will define what I mean by efficiency. This is the efficiency definition in writing. on the previous slide, and this is the picture of what we think efficiency should look like. There are 12 individuals here moving through this river, and 10 of them which are presenting downstream. Out of these 10, only 8 of them will find the entrance of your fish pass. So this number really defines the attractivity of your tool. That's 80%. And out of 8 of these fishes, only 6 actually go inside. 6 is the rate of the accessibility of your fish pass. And out of the 6, only 4 come out and go downstream. So this ratio of 4 out of 10 is the crossing rate. So 4 individuals came out of 12. That gives us the overall efficiency. It gives us the sum of the attractivity, accessibility, and crossing rate, which was 40% in this case. The RFID technology is a way to assess tools. It's the radio frequency identification. So this technology is passive, and the point is to get tags. These tags are not emitting a signal you receive. These tags respond to a stimuli. The stimuli is delivered by an antenna delivering magnetic field, and you can get a digital code to individually track each RFID. So the point of this passive telemetry is the fact that we don't have batteries in the tags, in the fishes. So they have unlimited lifetime, so we can do it over the long term. Other tags are miniature tags. They can be 9, 12, 23 millimeters, so we can tag fishes which are as small as 4 centimeters, so we can work with different sizes.
And it is not very expensive. It costs between 2 or 3 euros per transponder, so we can mark many fish. And it has an interest for the follow-up. I come back to that later on. So with this passive telemetry, we have got detecting distances which are limited, a few centimeters to 1 meter. So with the design of the antennas and where we are going to put them on the works or rivers, it will depend on what we want to measure. I've put a few pictures of the tagging, so you can see small crystal capsules. We put that in the peritoneal cavity of fish because it is a very good area. You also have on this picture where we receive the data received by antennas with batteries for isolated sites. Then, for the design of antennas, we can put that within fish works, so with frameworks like that. The detection is very good, and with new technologies we can have antennas design which are different, and we can investigate more. So this antenna is outside of the work. It is particularly interesting when you want to have data about attractiveness. You can go with your own antennas on the rivers 5 to 10 meters and to make detection between the obstacles. A last word before giving you examples. Regulations. We have got laws about the animal marking. So when you have an RFID project, the establishment should use animals for scientific purposes. The people doing the tagging should be trained, and you should receive a favorable opinion from an ethical committee on animal experimentation. So examples. The crossing at the level of a fish pass, ascending, so it is a successive obstacles. It is a device with 8 antennas, 4 of them at the entrance of the fish pass and 4 of them at the end of it. So we can have an accurate readingness and with robust data. And you have got a lot of data, and you need to be rigorous in the way that you are treating this data. For the marking, well, we take the fish, we make a small incision, we put the tag, and then after 3 months, you can see different levels of healing. 24% of the individuals marked were of less than 24 centimeters. We also marked Sophia and sculpins. For the movement, the displacements, we can define different behavior groups. In the first group was about fish that we never detected, so fish which died during the manipulation period, or they were fished maybe, and also fish which didn't find the entrance of the fish pass. And we couldn't assess the attractivity of the obstacle. Then we have got 3 groups with similar behaviors. They arrived at the antennas 2, 3, and 4, but they didn't go out of the fish pass totally. And the last group, which went out of the barrier, 16% for truth and 32% for the scalping. So it is important to mark many fish because we have got different behaviors between species and even within the same species. But there is a determinism of movement, 56% for the barbeau and 98% for the scalping. For the fish downstream, we have got level of crossings which are significant, 80% for the truth, for example, 70% in less than 1 hour and different sizes. That's all for the ascending fish, and now for the descending fish. What we can do with this technology on an hydropower dam with active telemetry, with contrast for the size of the fishes and also the cost of the tags. So if we can avoid hydroelectricity, is it efficient or not? So you have got different possibilities, as you can see on the screen with the images. We equipped all these exits of passes, and to know the migrating pathways of the fish, so you have got illustrating pictures. At the level of the fish elevator, we put some of them, and also downstream of the river. It was too complicated So we put these two antenna downstreams, and we had information about the fishes which took this way. In terms of results, we marked 937 fish. We had a few descending passages, and we had low flows during 1 year. Maybe it had an impact on the results. It is important to understand that we had 14% of the fish which were not detected in the descending canal. So the main fishes with descending, we needed to see efficiency of the exit of the fish pass. Now, quickly, we'll talk about the genetic tool. Simon Blanchet this morning talked about that. Here we are focusing on the assessment of the genes flow linked to the barriers, obstacles. It is an adapted tool for this kind of measures. It is an interesting tool because we can work on a bigger scale and assess different dams simultaneously, with cost linked to genetic analysis, which are decreasing at the moment. The first example, it is an operation organized with the Fish Federation of Saône-et-Loire. So we analyze the fragmentation of the area by obstacles, and on this river we had 3 impassable passages, one of anthropic aspect and one of natural aspect. And we wanted to have an initial aspect of it. So the methodology that we implemented, we cut the river in 8 parts. We realized 21 to 51 samples of truths, and we did genotyping of all that, and we obtained the genetic diversity in all stations and also the differentiation in terms of genetic structure between the different stations. That's what we have for the results. Here on the graphs, so here on the x-axis, for each line it is a truth, and the more it is colorful, the more you have a genetic diversity. So what we can see with these results is that we have more genes in downstream. It is due to the natural flow. So we have got an isolated population, a population which is intermediary and influenced by the isolated population and the downstream population, and in the background not a lot of genetic structuration due to anthropic structure. This is our conclusion. We didn't expect that on this notion of genes flow. We are responding to different issues here, and we need to integrate other issues. One of the limiting aspects of these dams, it is about temperature. We have got problems of temperatures on these streams with fish movement. And the heart of the problem was here. So we have got a quite interesting answer with this tool. Another illustration on the same theme and the same tools with the Liseron Basin. So a study with the Fish Association of Rhône. So from 2008 to 2014, it was a little bit complicated because we needed to do assessment on different dams. It was difficult to do it dam by dam. So the genetic tool was particularly relevant. But we didn't started the project, and the protocol is not optimal. You will see that with the repartition of the stations. You have got a scheme here. So some stations, we took the samples before the works, others after, or the two. So it's not ideal, but we have partial results which are interesting already. Same analysis with truth and 14 microsatellites. The results, it is the same way to read the graphs. We have got populations which are isolated. The population 1 and 5 suffering from geographical isolation and very few gene flows. And on this axis, we have got a genetic diversity assured thanks to input from upstream. So some works were done and we did some experiments again, and we realized that we had individual movements between the two areas, so significant results on the 8 and 5, a restoration of the communication between the populations, and we have got a few isolated sites in the black circles. Population 3 upstream in comparison to population 4, and a fish path was built with a problem of efficiency. And we need to go back and check that. And the last application, it is when we put the two dams together, we can focus the obstacles to treat and prioritize. This is one of the use of the genetic tool for assessment. As a conclusion, you can see that we have got different tools and we can obtain relevant data that we can measure in situ. These different tools, well, it is complex for the project manager because you need to make the good choice. So here, an information table. It is interesting for you. You have got the advantages and disadvantages of each tool and what you can do with them. Generally speaking, what we can remember is that these tools they have different applications and the possibility of operational answers to the questions. The interest of all that is to multiplicate the feedback. Normally these tools, they are only for research firms or pilot sites, but there is a big interest and we can improve the know-how and have better rehabilitation for ecological continuity. To strengthen our know-how, to feed public debate and contribute to the improvement of acceptability of the RCE policy, there is an interest of transfer of know-how from the scientists to the managers. And the managers will have better knowledge and know these tools and know what we can do with them. So a highly collaborative method between managers and scientists, and to use them at a bigger scale. Once again, I thank the people who helped us to work on these different examples. I thank you for your attention as well.